All right, so this is part 2 of chapter 11, genetics and evolution. It's mainly genetics, but a little bit of evolution. So last time in part 1, we talked about twins. And twins can come from one egg or two eggs. If it come from one egg only, this is going to be the identical or monozygotic twins. And if this is coming from two eggs, can a lady like ovulate too? Yeah, sometimes it happen. Most of the time, it's one that's ready for fertilization, but sometimes, occasionally, it can be two. And as I mentioned, because of there are a lot of sperms that's available, millions. So some of them, if they found two eggs, they were fertilized two eggs. If they found three, if they find three, they will fertilize three, and so on. So it's all about how many eggs. One egg, it will give you the identical the identical twins, and those identical twins will be having shared placenta. So when they cut the placenta, both of them are disconnected to the mom, or their circulation is disconnected with the mom, and it is one placenta only. This is for the identical or the monozygotic twins. For the fraternal or dizygotic twins, as I mentioned, it's two eggs. So obviously, two sperms, the strongest, will fertilize, and this will result in two placentas. So for the fraternal one, or the dizygotic or unidentical twins, um, they studied the reason for that. Why would a lady or a female produce more than one egg, even though it's normal that there should be one oocyte or one ovum? They found that there is a gene on her X chromosomes, one of their X chromosomes, that cause something called hyperovulation, which is ovulating more than one egg per cycle. Normally, it's one egg per cycle. But if some ladies have this gene, she can produce more than one, and she can take it from the lady specifically can take it from either of the parents. But it's not like the only thing, and it's not that strong. It doesn't has to be there. Just a little bit more information about it, but I do want you to understand the difference between identical monozygotic and fraternal dizygotic, which is unidentical. One ovum with shared placenta, two ova with separate placenta. This is the most important thing. Next part is, if you guys still remember when we talked about the genome or the genes, we talked about the 23 pairs. And I will remind you that from pair one, so it's two together, but it's two homologous chromosomes. But from pair one until pair 22, these are called autosomes. It is or autosomal genes or chromosomes. These chromosomes are responsible for everything in the body except uh, sexual reproduction. They are not responsible for sexual reproduction and not responsible for producing male versus female. It's not responsible for sex. Number 23 is the one that's responsible for sex, determining the sex of the baby. So this is obviously a female because this is X chromosome and this is X chromosome. How did I know? Because X chromosome is a very big chromosome, and both of them looks exactly the same. So obviously this is a female, while this is a male. How did you know? Y is very small. So if you see this big X and there is a tiny one beside it, this tiny one is the Y. So obviously this is a normal female and this is a normal male. Why are we talking about that? Because there are some traits and some diseases or abnormalities that are going to be based on our understanding of this right here. So the bottom line is there are some traits and conditions that are linked to genes on the X chromosome, and we call those X-linked traits, which is what we're talking about right now. And very few are called Y-linked traits, and this is because it's small, it contains less genes, so obviously less linked traits. So X-linked traits and Y-linked traits. X-linked traits, including the abnormalities or the syndromes or diseases, this is a lot more common than the Y. So what's happening here simply is they noticed that there are some traits. In this case, it is diseases like hemophilia and color blindness. They noticed that it is more common in males. And what's the explanation of that? The explanation is males have one X chromosome and one Y chromosome, while females have two X chromosomes. So what? So this is dizygous, two X's, meaning that if there is a gene at this point right here that carry a condition, let's say hemophilia, there is a correspondent gene on the other one that can be normal. And if that's the case, you have two copies of this gene. One of them is bad and the other one is good. The good is going to mask the bad so it doesn't appear in females. While in males, if there is, I'm talking about the X link now, on the X chromosome specifically, if there is a bad gene right here, remember, I'll remind you again, chromosome, DNA wrapped around histones, chromosomes, we're talking about chromosomes, DNA, and in the DNA, each part of it is gene, 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 gene. There are a lot of genes here. So let's say this gene right here is a bad gene. In this male, there is no other X chromosome. There is Y. So there is no other X chromosome that can mask it. So obviously it's going to appear. One bad gene is enough to give you the disease or the trait or the abnormality. And that's why we say hemophilia is a lot more common in males than females. Because it's enough to have one gene. You get it from your mom, for example, in this case, you got it. That's it. Why don't you get it from the dad? Because this is male. If you got this from your dad, how can you get this? Where are you gonna get this? So if you got the Y from your dad and you got the X from your mom, and if this one is the one that have the bad gene, there is nothing else to mask it, so you get it. And here is the explanation why we say X-linked traits are more common in men, and it go to them to the male from his mom, not from his dad. Okay, I hope that's understandable. And as usual, if you have a question, stop me anytime you want. I want to make sure that you guys understand this. So that's why it is more two important pieces of information. It's a lot more common in males, and males get it from their mom, not from their dad, because the dad give the Y if you are a male, and the mom give you the X. But the males have X and Y. I understand, but how are you a male if your dad give you the X? How can you get the Y? No, you get the Y from your dad, you get the X from your mom. This is how it works. So we call that hemizygous. Males are hemizygous. Hemi means half. So they get half, which is one X from their mom, and the other one and the other one is a Y from the dad. So obviously, if we're talking about X-linked recessive, males are going to get it. Just get one gene and you're done. But females are not going to get it because If there is one here bad and one good, uh, uh, she will not get it because the good is dominant, the bad is recessive in this case, and it's going to mask it. So female cannot get it at all? No, they can. If this female got bad gene from the mom here on the X chromosome, and another bad gene from the other from the dad, because this is female, she will get X from the dad, X from the mom, and X from the dad. So if it happens that this dad has the disease, like in this case, there is a bad gene here, and if it happened that the mom is at least a carrier, you mean the mom has the disease? No, I didn't say that. The mom can be a carrier. She have one good and one bad. But if this unlucky female got the bad one out of the two X chromosomes from the mom and get the bad X, which is only one from the dad, she will be affected. But obviously, it's a lot more common in males because of that reason, because one is enough. You don't have the other one to give you the chance to mask. So if you look at this Punnett square right here, let's say this is a female that has XX, obviously, and this is a male that has XY, which is chromosome number 23. And this one of the X's is okay, is fine, normal, and the other one is bad, let's say hemophilia. So it's X-linked recessive. Does this lady or mom have the disease? No, she does not. Why? Because this lowercase, which is recessive, is bad, but she have another X that has a good chromosome. So it's going to mask it. So this lady, if you see her, does she have any issues? No, she's completely normal. But if you do genetic study, you will find that she is actually have one of her X chromosomes that has the issue, but it doesn't show up because the other one is masking it. So what do we call this lady or mom? Carrier. What's a carrier? Meaning she has the gene, pass it off to the next generation, but she's not affected. Just a carrier. I take it from here, send it there. From my parents to my kids, but I'm not affected. I'm just carrying it. In this case, this is a completely normal dad. His X chromosome is okay, and his Y chromosome, there is nothing there because it's a Y chromosome, and we're talking about the X-linked traits, meaning that the bad gene in this case is on the X. So what are the probabilities? This one got one good and one good from the mom, one good from the dad. He's completely normal. This one has one good from the mom and a Y, so there is no issue. This girl, she got one bad from the mom, I mean from the dad, and one bad from the mom. She got the bad one. So she is a carrier. Is she going to have the disease? No, she's just a carrier. She will pass it on to her kids. And this unlucky one right here, uh, he's going to be affected because he got one bad from the mom, and that's enough because the Y will come from the dad. So what are the chances here? It's 25%. But what's the chances of the males? If you see here, two of the males have it, 50% chance. Here is the Punnett square. It's just explaining the same thing right here. You have a carrier female and a normal male. This is what we just talked about. We'll give you normal female or normal girl, carrier girl, normal male, affected male. And this is the pedigree. So these are the two ways of predicting the outcome or predicting what's going to happen later. Does it give us like 100% confirmed information? No, it's prediction, probabilities. If you go to the doctor and tell him, what are my chances? Like my wife is a carrier, or the mom is a carrier, and the dad is okay. What are the chances? The chances only, not confirmed, not 100%, chances. So you can do Punnett square, or you can do the pedigree, which goes generation after generation after generation, vertical and horizontal, and we talked about that before. So we can just predict. So here, if it is completely blue, this is a normal color. If part of it is black, white like this, she's a carrier. If it is completely black and white, this is affected. So if this is a normal male, which is this one right here, but we do it in the form of pedigree, and this is a carrier female, which is this female right here. Look at those four, and instead of putting it in a square, you can spread it like this. Like this one here is the affected one, which is this male right here. And this is a completely normal male, which is this, followed by the females. One of them is completely normal, which is this, oops, which is this one right here. And the last one is a carrier, which is this. So what are the chances if I'm coming to you and asking you, I am completely normal, I don't have any issues in my genes, but the mom is having, she's a carrier to hemophilia or color blindness, for example, X-linked recessive. Can you tell me what the chances for my kids? I will tell you, yes. Uh, if you get a male, 50% will have the issue and 50% will be completely normal. If you have a female, 50% she will be completely normal and 50% she will be a carrier, not affected. And you can do the same. So this daughter uh, is matched with another male who's normal, you get the same chances and so on. So this is what we talked about so far. And this is the most important part of all what we talked about. Punnett square and the pedigree, you can have an idea about it. The minimum is just to understand how does it work. Like, not going to give you like a detailed pedigree or something like that. That would be too much for this course. But at least you can be asked about general idea. What is it? What is it for? So which trait or traits are affected by many different genes, different genes. That's going to be the polygenic, because poly means many, gene, here is many gene. So this is polygenic. The other one is which traits are shaped by more than one gene and this is important, environment. If the environment is there, this is multifactorial. Genes is a factor, environment is another factor. We call that the multifactorial. This is important to know. Okay, this is the core of the chapter, this slide specifically. What is the expression of one gene that affects more than one trait? This is called the pleiotropy. Pleiotropy, this. One gene give you multiple things, like Marfan syndrome that we talked about before. One gene is bad, but this child will have issue in his spleen, issue in his bones, issue in his joints and the skin. So it's giving you multiple things. We call that pleiotropy. And do not confuse multifactorial from pleiotropy. Multifactorial, genetics is a factor, environment is another factor. Pleiotropy, one gene, give you more than one issue in different organs. What kind of dominance is the expression of combination of both? So both are expressed if this is a heterozygous, like the wavy here. Remember, we talked about the dominance. There is complete dominance, incomplete dominance, and codominance. Which one that both of them will be expressed, like wavy hair coming from a mom with straight hair and a dad with curly hair. She will be something in the middle. So obviously both of them um, are expressed. Um, so this is called codominance. No, I mean incomplete dominance. I'm sorry, incomplete dominance. So both of them will show up. The other example was this flower. One is white, one is red, and the result is pink. What do you call it when more than one allele uh, is fully expressed, not just incomplete, fully in heterozygous, like blood type? This is a codominance. So this is incomplete, and this is codominance. Last one is, what is the percentage from 0 to 100% that the gene can be expressed? This is obviously penetrance. Penetrance meaning penetrate, which is the genes will go to the next generation, or sometimes it doesn't go all the way. Now, evolution. We need to know that all humans first was in Africa. Every single person on Earth, the ancestors were in Africa. And this is about 60,000 years ago, where they started to migrate out of Africa and moving to the neighbor, what we call it now countries, but it was just a land, moving from Africa to the beginning of Asia and just a little bit of Europe, crossing the Mediterranean Sea, just going on the neighboring lands, and they start to move. Migration started until they inhabitate the whole or most of the earth. Um, and they did mapping of the appearance and frequency of genetic markers to know how the ancient world looked like. Um, what's the scientific evidence? How did they find out that these are all coming from one? Because the DNA of all human beings are alike. So if this person is completely different than this person, this is uh, black or brown, then short and black eye, and this is white and tall and yellow hair and whatever, completely different. If you if you studied their genes, you will see it 99.9% the same. 99.99% the same. So it's 0.1% that makes all these differences. But otherwise, it's 99.9%. This is the most important one. Also, they studied the fossil for evidence. Like they get these rocks and different types of fossils, and they found bones, and they look at the bones, it looks different, and that's why they gave us this theory of the ancestors, where they started and how did they look like, and so on. They also found that the behavioral pattern is the same. Like every single person on Earth, they either listen to music, play music. Yes, it is different music, different art, uh, burial, shelters, and all that stuff. It's in common. It's behavioral pattern. Yes, everybody's doing it in his own way, but it's in common. There is also something called mitochondrial DNA. Remember the DNA, we talked about it in the nucleus. But if you still remember, we said it's not only in the nucleus. Like this is the normal location, most of it. But there are some DNA in the mitochondria, and this DNA from the mitochondria will go from the mom to her kids. So they can track that and study it to know the ancestry. So what's the definition of evolution that you have to know? Evolution is the change in frequency of an allele over time. This allele is for dark skin. Everybody has a dark skin. And the frequency started to decrease, and there started to be some lighter colored, obviously, because they migrated to like colder places. So the frequency start, instead of 100%, it start to become 90%, 80%, 50%, and so on. So it is decreasing. If there is an allele for the beginning of the tail, for example, and then it disappeared, and so on. And what's happening here, the theory behind that is natural selection. The nature selects which allele to stay and which allele to disappear. Depending on what? Natural selection. This is an interaction between this allele and the environment. If this allele help you to survive in a cold weather, this is going to continue, and other people who don't have this allele will not be able to resist, so they're going to die, and those who are selected to continue will carry this gene that protected them, and so on. This is called natural selection. When we say this trait is selected for, it means it become more common in the population. There are some traits, we call them advantageous traits. Advantageous means they give some individual advantage to survive, and when they survive, they reproduce. So those people are selected naturally because they have advantageous traits. They will be favored, they will be picked by the natural selection and become more common in the population. And the unsuccessful traits are going to disappear. And that explains the disappearance of certain species. And like if you look at the fish, we have different hundreds, maybe thousands type of fish, and others disappeared. Where are these? They didn't have this advantageous trait. They were not naturally selected. They disappeared because those who have the advantageous traits, for example, if this fish used to live in this area and this area now is cold, those who have the allele, the advantageous trait because of the allele, will be able to deal with this situation, and others are going to disappear. So those who are selected, they have the advantageous trait, they will continue, they will live, survive, reproduce, and this trait will become more common in population, in this case, in the fish population. This is one thing. The other thing is some traits, even though these are not advantageous traits, these are, we can say, like bad traits, mutated traits, unadvantageous, non-advantageous, like it's bad traits, but they can still survive. We're talking specifically here about, in this example, about humans. So in humans, there is a disease that's called sickle cell disease. And there is a gene in those people's DNA that's bad. There's a bad gene. Mutated means bad. It was okay, normal, but it got mutated, so it become bad. And this disease is homozygous condition, means if you have the two pairs, the two alleles in the two chromosomes. Remember the recessive homozygous? So if you have it, you have the sickle cell disease. But in this case, and this is something that they noticed, carriers, unlike what we talked about before, before we said, if you're a carrier, you don't have any issues, like hemophilia. If this female is a carrier, she has one dominant and one recessive, she's completely fine. You don't even notice anything, except if you studied the genes, but she will pass it on. So this is a carrier. They found something here interesting. In this disease specifically, sickle cell or this trait specifically, they found that the carrier is not 100% normal. In this case, they found something that's very unusual and very weird. They found those people to be protected against malaria for some unknown reason. And this is something additional that I just want to tell you that with sickle cell disease, look at the normal RBCs here. It looks like this, circular but not spherical structure. There is a concavity here. And it is flexible, and it goes like this because they flow, red blood cells flow in the circulation, and they go in different vessels, arteries, and veins, and so on. But the capillaries, the tiny ones, the diameter is almost the same as the red blood cells. So if the red blood cells were not in this shape, they will be stuck in the capillaries that's too tiny, but they are flexible and circular like this, biconcave. But look in this case, how the RBCs looks like. It looks like a sickle. Look at this. What's bad with that? What's bad is those red blood cells can flow freely in the circulation. They can pass through the capillaries without issues, and they don't come together because, like, if you try to get two balls, two soccer balls, two basketballs, whatever, and try to put them together, if you push, they will go away from each other. You cannot put them together, they cannot stay. But in this case, one is like this and the other one's like this. They are easy to stick together, making blood clots. They are easy to be stuck in the capillaries and get destroyed. They get burst, we call it burst. Like here they are started to burst. So if those people have the actual sickle cells, have the sickle cell disease, which is homozygous, they get the actual condition, and once the oxygen goes down a little bit, the red blood cells start to break down and they get the crisis. But the carriers just have a little tiny thing which is protected against malaria, and also if it is very, very low oxygen, they might have a minor attack. So next is genome sequencing. Can we do sequencing of the genome? What's a genome? Genome is studying of all the genes. Genome, all the genes, the whole complete set of genes. We call it genomes. Can we do sequencing and studying it, mapping it from the DNA, get a DNA and do mapping and studying of the whole entire set of genes, which is called the genome? If you do that, you will find there is every single human being has his own unique DNA. We call it the unique blueprint. And that's why if you do DNA analysis, they can tell that like this kid, uh, if you match it with the dad, they can say, no, he's not. It's impossible. Because they compare it and they do mapping of both of them and they compare it, there is a way to tell. But if you use that mapping to give you the exact ancestors, I know that some of us might have done this genome studies, and yeah, my grandparents came from whatever, Italy or Germany or China or any country. Yes, but they cannot tell you exactly the ancestors, like track it all the way down, because um, it is not that accurate. Because they compare and there can be some difficulties in the comparison. But the good thing about the genome sequencing is they can tell you the risk of having a disease. If you tell them, do I have a risk of having cancer? Cancer of what? Like testicular cancer? Let me take a sample and I'm going to study it and see the mapping. and compare it to somebody having this disease. And I can predict, I can tell you not 100%, but you have a good chance of having it. But I cannot tell you, yes, you have it, or yes, you will have it, or something. It's not that accurate. So which country did we evolve? Obviously, Africa. Evolution um, is the chances, the change in frequency of, very important, allele. Natural selection depends on the interaction between, obviously, the alleles and the environment. When you say this trait is selected, it become more or less common. Of course, it is selected because it is an advantageous trait. So it's going to be more common, and the unadvantageous is going to disappear. People having or with homozygous or heterozygous sickle cell are protected. Of course, heterozygote, which is weird. Homozygous will actually have the disease. They are not protected against malaria. They have the disease. True or false, genome sequencing can identify genes that predict when you will contract a given disease. No, it can only predict, but it's not exactly. It can just give you you might have a chance or not. Okay, and as usual, these are the summary slides. This is what we already went through. So I'm not going to repeat it again. It's the exact same thing that we did. So I'm going to stop sharing now. And this is the end of this chapter.